Assalamualaikum students, it's the second part of iron metabolism. In this video we will discuss about regulation of iron metabolism and its absorption. Basically, iron is regulated in our body at two stages. First, at mucosal uptake, when iron is absorbed from the intestinal lumen through the intestinal cells, and the second is during transfer from the intestinal cell to the blood, or from the liver cells to the blood. Hepcidin is the master iron regulator which is produced and released by the liver. Most of the iron in our body is regulated by hepcidin, which is a protein in nature. Main function of hepcidin is blockage of ferroportin. Ferroportin which Ferroportin play an important role in the transport of iron from the enterocyte to the blood, from the liver to the blood. Ferroportin transporter present on the surface of enterocytes. When the enterocytes absorbs iron from the lumen of the intestine, the ferroportin then release the iron to the bloodstream. When the level of iron in the bloodstream rise, it stimulates the liver to release hepcidin. Hepcidin then block the ferroportin to stop the release of iron to the blood. Thus, the level of the iron in the plasma is regulated. As the release of the iron from the enterocyte to the blood is stopped, now the level of the iron in the enterocyte is increased. Now here is the need to stop the absorption of the iron from the lumen of enterocyte. Now hepcidin from the liver targeted the enterocyte to stop the absorption of iron from the lumen of the enterocytes, and thus iron absorption from the lumen of intestine is regulated. Hepcidin can also block the release of iron from the liver. Spleen macrophages play an important role in the restoration of iron. The abnormal RBCs that circulates to the spleen and is digested, and iron is released. The hepcidin from the liver block it. Here is a question: what stimulated the release of hepcidin? What are the factors that cause the release of hepcidin? There are a number of factors which are involved and that cause the secretion of hepcidin. Number first, inflammatory cytokines that are released by macrophages. For example, interleukin 6, which can cause the release of hepcidin. The interleukin 6 from macrophages cause stimulation of the liver cell to release hepcidin. The second factor is the level of iron in the plasma. When the iron level in the plasma increases, it will stimulate the liver to secrete hepcidin. The third factor are the bacterial or pathogenic lipopolysaccharides, which can stimulate the liver to secrete hepcidin. The main regulator of the hepcidin release are the HFE protein. HFE stand for high iron. H mean high and Fe is the symbol of iron. HFE protein is coded by HFE gene, which is also known as hemochromatosis gene. HFE protein interact with other proteins, and thus the iron absorption and metabolism is regulated. How iron is regulated at the stage of mucosal uptake during absorption from the lumen of the intestine to the enterocytes. At this stage, how iron is regulated. According to recent modern studies, it shows that here is a factor HIF2 alpha protein, which are the mediator of cell adaptation, which are coded by a gene. These proteins can cause the enterocyte to adapt their cells according to the condition to increase the absorption of iron or stop the absorption of iron. The HIF2 alpha proteins are coded by a genes which is called SLC SL SLC11A2 gene. This gene can code the protein which is an important transporter, DMT1 divalent metallic transporter 1 protein, which can cause the absorption of